Welcome you all in today's class. Our today's topic is cell division. This topic is from the class 11 Maharashtra State Board biology book. Myself Sudhir Gadge. Let's look at the introductory part of this topic. Okay. Inside our body, one major important process which is taking place is known as wound healing. Okay. This wound healing process occur after any cut or injury to our body. Okay, this process require uh, few days, few days to many months or some months if deeper injury comes. Okay, behind the wound healing process, one important process is cell division process, formation of new cells. Okay, after cutting to our finger, one process immediately happen known as a hemostasis, means mm, stopping of bleeding. Okay, after completion of hemostasis, our finger become inflamed, that is become red, hot, and uh, large in size because of inflammation. Why this happen? Because in cut part of our body, there is a chance of entry of some viruses, bacteria, especially bacteria, okay, which causes skin infection. Hmm? To avoid this, our natural immune system will come and uh, it causes inflammation. Here, gathering of cell takes place, particularly white blood cells. Okay, and because of that, this area become red and inflamed. After completion of inflammation, next process which will happen known as proliferation. That means rebuilding of new tissue. This process occur after some days of injury. Okay, here you can observe the healing process. The healing process takes place because of in cut area new cell will form, and that new cell build up, and after a few days we will observe our finger become as it is. So this is a wound healing process. Why this process takes place? Because of uh, to get healed from the injury, and one major important role here which is cell division. What is the difference between growth of non-living material and living organism? Okay, uh, in our biology book, first topic is the living world. Here uh, we have seen living organism have some important characteristics which are known as metabolism and growth. Metabolism and growth. Okay, here we have observed already grow in living organism, growth is from inside to outside. Okay, we cannot observe outside growth in living organism. Growth, it takes place from inside to outside. For example, increase in number of RBC, increase in number of WBC, wound healing process, these are all example of internal growth. Okay, but in case of non-living organism, this growth takes place from the outside. जैसे कि if there is a any stack of sand, okay, because of deposition of new sand, this sand layer, this here we can observe a growth of sand. Okay, this growth takes place from the outside. Okay, so this is the best example of growth in non-living organism. Okay. Second, metabolism. Living organism perform metabolism. Metabolism means reaction. Okay, inside our body, to get energy from food, uh, our body do some reactions. Okay, which are known as metabolic reactions. This is a main characteristic of any living organism. We people produce energy by using food. Plant produce energy by using photosynthesis. Okay, this energy is required for the life processes in plant or animal, and which include some reactions, and reactions are come under the metabolism. Okay, next main important feature is the organization and changes. Biological growth is highly organized process, is directed by DNA. Okay, this leading to maturity and development. Non-living matter increases randomly without the biological development. Okay, so biological development direction under the direction of DNA is a main important feature of any living organism. Okay, let's see some introduction. In case of plant and animal in living organism, in case of we people also, our life arises from a single cell. Our life is arise from the single cell. Okay, uh, during the during some period which is known as pregnancy in case of animal. Okay, this cell undergo multiplication. One cell divide and produce two cell stage. This two cell stage in next few days convert into 8 cell stage. Then 16 cell stage, then 32 cell stage, and so on, and finally multicellular organism will produce. Okay, this is a tendency of any living organism. Initial cell, which is known as a zygote. Okay, and from that zygote, uh, multicellular organism is produced. This is the main important feature of living organism. In past, cell theory suggests that every new cell arise from the pre-existing cells. So I can say this is a mother cell. This is a mother cell, and from this mother cell, new daughter cell will form. So this is a pre-existing mother cell, and from this mother cell, new organism will form. For example, uh, we people we people born to our father and mother. Okay, so we produce from the our mother and father, which are pre-existing cells. This is the best example of this theory. Every cell is arise from the pre-existing cells. uh, Cell theory also suggests that all living organisms are made up of cells. So हर एक organism के अंदर cell होता है. Okay, you can take any living organism, plant, animal, uh, bacteria also. They contain cell. Second theory suggests that cells are basic unit of life, as like our home. Hmm? In case of our home, small bricks are present, and by using that small brick, uh, our home will form. Okay, like that, our body is formed from the single cells. So single cell is a functional unit of our body. Okay, or which is also known as the basic unit of our body. The brick is a basic unit of our home. Like that, cell is a basic unit of our body. Okay, third point in cell theory suggests that the cell arises from the pre-existing cell, as I already discussed. Okay, every living organism is formed from their mother. Okay, which is a pre-existing cell. Hereditary, we get some genes from our parents, mother and father. Means gene flows from mother and father to their offspring. Means heredity information is passed from one cell to another cell. This is a justification of the theory. Okay, then inside our cell, some chemicals are present. You can look at the biological membrane. Biological membrane is a lipid bilayer. Okay, lipid bilayer. Lipid, these are the chemical compound. In this bilayer, cholesterol is also present. So cholesterol is a chemical. In this bilayer, phospholipids are present. Phospholipid is one of the chemical. Then some proteins are also present. Proteins are chemicals. So according to theory, all cells have basic chemical composition. So in our biological membrane, lipid, cholesterol, phospholipids, they are present in certain composition. Okay, last point in the theory suggests that the energy flow occur within the cell. Okay, so mitochondria is a powerhouse of cell. Inside our cell, mitochondria generates energy, and that energy is flow from mitochondria to other part of the cell, or we can say to other organelle. Okay, let's look at the cell cycle. Why cell cycle? Because cell produce new cell. This is a mother cell and this is a daughter cell. Mother cell produce daughter cell, and this process is a cyclic. Why? Because this daughter cell again produce new cell. This newly formed cell again produce new cell. Okay, so this process is um, uh, continuously operating. That's why we here we have written here cell cycle. Okay, this is a by definition it is sequential event occurring in the life of cell is called as the cell cycle. So cell cycle involves some sequence we will discuss in later part. Okay, cell cycle is a sequential event, means it includes steps. After completion of one step, cell goes to next step. After completion of this step, cell will go to next step. So this process is a sequential, takes place in a sequential manner. Okay, here every step is very important. In general cell cycle, these two main phases are very important. One is interphase and another one is a M phase. Okay, so in every cell cycle, two important phases are present. One is interphase and another one is a M phase. Interphase माने तो this is a preparation. I can say this is a preparation. In interphase, cell prepare for future division, and M phase माने तो mitosis and meiosis. Mitosis and meiosis. Okay, in interphase we can observe cell undergo growth or sometimes cell take a rest before it undergo future division. So here I say it is a preparation for future division, and in case of M phase, this is a period of division which divided into mitosis and meiosis. You can observe here, this is a complete cell cycle. This is a cell which is present at the start of cell cycle. So as I already told you, cell cycle events are divided into interphase and M phase. From this point to this point, up to this point, this is a interphase. This is a interphase. Okay, means phase of preparation. Why preparation? Preparation for future division. Preparation for future division. So interphase is divided into three parts. One is G1 phase, another one is S phase, and third one is a G2 phase. Which phase? G1 phase, S phase and G2 phase. G माने तो growth or gap. Growth or gap. So this is the first growth or first gap phase. G2 माने तो growth 2, second growth phase or second gap phase. Second gap phase. S माने तो synthesis. S माने तो synthesis. Okay. So during first growth phase, this is the first step from this point to this point. Okay. Here cell prepare itself. Cell undergo growth, means consider this is the first cell. This is the first cell. Okay, which is in future undergo division. Undergo mitosis, which will undergo mitosis in future. Okay, for future division, cell during a G1 phase, cell undergo some growth. Here cell increase their size. Cell increase some organelles present in there for future division, because यहाँ पे क्या होने वाला है, in next step cell will divide into two cells. Okay, so for that purpose cell in this G1 phase increase organelle number. Okay, inside the cell, or different type of organelles are present, like mitochondria, chloroplasts, lysosomes. Okay, these organelles undergo duplication during G1 phase. Okay, some proteins, some RNAs also synthesized during this G1 phase for future division. Or basically, during a G1 phase, some proteins are synthesized or enzymes are synthesized which are required for next step or upcoming step, which is a S phase. Okay, duration of G1 phase is 8 hours. 8 hours. Or sometime it is greater than 8 hours. After completion of G1 phase, uh, cell will cell may go to uh, S phase. S phase involves synthesis. Synthesis of what? Synthesis of DNA. Here, mother cell produce new DNA. Mother cell produce new DNA molecule. Okay. Initial mother cell, only one DNA is present. Okay, during S phase, mother cell produce another copy of DNA, so that now the cell contains two copies of DNA. So this will happen in S phase. This phase takes 6 to 8 hours for completion. After completion of S phase, cell move towards the next phase, which is growth 2 phase or gap 2 phase. This process takes 2 to 5 hours for the completion. During this process, again cell undergo growth. uh, Cell synthesize some proteins, some factors, growth factors, which are required for the upcoming mitosis or meiosis process. Okay, this process complete within 5 hours, 2 to 5 hours. So in all, G1 phase, S phase and G2 phase, these are the part of interphase. Overall, uh, approximately they want sorry they need 21 hours for the completion of G1 phase, interphase. Okay, they take 21 hours for the completion of the interphase. After completion of interphase, cell may go towards the mitosis. In mitosis, uh, we can divide mitosis in four different steps. One is prophase, metaphase, anaphase, telophase. Okay, you can observe mitosis is a continuous process, and that process divided into four steps. Okay, prophase, metaphase, anaphase, telophase, and cytokinesis. We can observe in interphase process. In interphase process, cell complete its nuclear type of division. Means in any cell cycle, two events are present. One is karyokinesis, and another one is a cytokinesis. So in interphase, DNA synthesis, copy of new DNA is formed, but this new copy of DNA distribute in two different cells. Okay, original DNA distribute into two different cells during karyokinesis process. This karyokinesis process complete in mitosis. Okay, after completion of mitosis, after completion of uh, nuclear division, cell may go to cytokinesis, means cytoplasmic division. Okay, cytoplasm is divided. Cytoplasm is will be distributed between two newly formed cells. So first phase is the interphase. This is a, we can observe here, interphase required 21 hours, which is very huge time. Okay, this is the longest phase of the cell cycle. This stage, interphase stage, is present in between two successive division. This is a mother cell. Okay, this is a newly formed cell. Okay, So mother cell after completion of interphase undergo mitosis or meiosis and form new cells. Okay, these two new cells. These two new cells again uh, spend their time in interphase and form two new cells again. So this is a interphase is a phase present in between two successive cell division. Okay, cell is highly active during interphase. We can observe proteins or cellular organelles, DNA, RNA will form. So this is a very active and busy area. Hmm? So cell is highly active and prepare itself for the upcoming mitosis. Okay, the interphase is divided into G1 phase, S phase and G2 phase. You can observe here in G1 phase, cell is highly metabolically active and duplicate its all the organelles and cytoplasmic component for the future division. After completion of G1 phase, okay, cell decide to go for cell decided to go for the copying of own DNA, which is known as DNA replication, which happen in S phase. After completion of S phase, after formation of new copy of DNA, cell will decide to go for G2 phase or second growth phase. Here, certain enzymes, certain proteins synthesize. Centriole undergo duplication in this phase. All this process takes place for the preparation of upcoming mitosis phase. Then mitosis will come, and in mitosis, these phases are present: prophase, metaphase, anaphase, and telophase. Here you can observe interphase, G1 phase, which is the first gap phase, okay, which start immediately after the any cell division. It involves formation of mRNA, RNA, tRNA, proteins required for upcoming S phase. Then then S phase will come. In S phase, DNA undergo duplication. New copy of DNA will form, which is known as a replication. Here, in this process, histone proteins get synthesized. Histones are the protein which require for the DNA wrapping. So, consider this is your DNA. After formation of DNA, it bind with histone protein. Histone stabilize the structure of DNA, newly formed DNA or existing DNA also. Then interphase. In interphase, next phase is G2 phase. Here, cell increase its volume, cytoplasmic volume. Here, again, cell metabolically active, very active. Here, cell produce new RNA, new proteins. Okay. Here, centrioles, which is a major portion required in mitosis or meiosis, which undergo duplication. Then M phase or period of division, where M stand for mitosis or meiosis. Okay. During the M phase, nuclear division takes place, means DNA is distributed in two different daughter cell from their parent, and after completion of nuclear division, cytoplasmic division also takes place, which is known as the cytokinesis. Karyokinesis is a division of nucleus, while cytokinesis is a division of cytoplasm. Okay. This process is very important because both nuclear divisions are uh, must for the cell division, because each cell require DNA copy. Also, after completion of this, cytoplasmic division is also very important because each newly formed cell require its own cytoplasm. So both processes are important. Okay. Then types of cell division. Cell divide in different manner. Either amitosis type means direct division from existing cell, new cell is directly formed without cell cycle. Okay, this is known as amitosis. Or mitosis or indirect division. Okay, indirect division might here haploid or diploid cell produce this type of division performed by haploid or diploid cell. And meiosis, this is a reductional division, only diploid cell produce. Okay. In case of mitosis, okay, two types of cell will come, either diploid cells, diploid cell, or 2n cells. What is mean by diploid or 2n? Means here uh, two copies of the DNA are present. Two copies of one DNA are present. And in case of haploid or n type, only one copy or single set of DNA is present. Okay, so both type of cell, diploid cell or haploid cell, can undergo mitosis. Okay, but in case of meiosis, only diploid cell having two sets of gene undergo meiosis. Only only diploid type of cell undergo meiosis. This process is known as a reductional division because after completion of meiosis, two haploid cells will form. Okay, the original cell contains two copies of DNA, but newly formed cell contain only one copy of DNA. Means this is a reductional division, reductional kind of division. Okay, see the example. In case of honeybee, drones which are haploid, you can observe here. In case of queen, which is diploid, having 32 chromosomes. Okay, from this queen, if fertilization takes place, okay, new worker honeybees will form having 32 number of chromosome, and from the unfertilized egg, drone will form. Drone contains 16 cells. From this drone, male will form. Okay, so here haploid to haploid cells are formed with the help of mitosis. Similar example, in case of Markensia, this is a bryophyllum. Here haploid spores produce gametophyte by the process of mitosis. You can observe here, these are the haploid spore having single set of gene. By using this, male gametophyte will form with the help of mitosis. Then first process, which is amitosis. Okay, it is the simplest kind of cell division. It is a very simple type of cell division. During the amitosis, this is the original nucleus. This original nucleus undergo elongation and means kind of stretching. Undergo elongation. Simultaneously, cytoplasm also undergo stretching. Deep furrow is formed at the center. Okay, after some time, the stretched nucleus undergo division and two new nucleus are formed, and same time stretched cytoplasm move to inside and two cells will form. This is the simplest kind of division where one cell convert into two cells. Parent cells convert into two new daughter cells by simple mean, just by division. Like consider this is a cell. If I cut this cell from the middle, two new cells will form like this. This type of division taking place inside the during the amitosis process. Okay, this type of division happen in bacteria. For example, E. coli. E. coli multiply, produce new E. coli within a 20 minutes of period. 20 minutes. Okay, in during a 20-minute, E. coli form new cells just by a mitosis process, means by the body division. This process also taking place inside the old cell or fetal cells, fetal membrane cells. Then mitosis. Okay, in this process, This is a parental cell which is 2n in number or n in number, diploid or haploid. Okay, parent cell first undergo prophase. Okay, during the prophase, chromosome undergo condensation. Chromosome sometime chromosomes means DNA is visible, and spindle pole formation takes place. Chromosome attached to spindle poles. Okay, after completion of prophase, cell will go to metaphase. Here all the chromosome align at the equatorial region. Okay, after completion of metaphase, cell will go to anaphase. Here cell completely move towards the pole, DNA completely move towards the pole, and after completion of anaphase, cell move to telophase, where complete two new nucleus will form, and cell karyokinesis is completed, and after that cytokinesis will happen, means cytoplasmic division will takes place and new daughter cell will form. Okay, after that cytokinesis will come and complete new formation of two different cells will happen. Okay, this is a brief introduction of mitosis. In future lecture, we will study in detail mitosis and meiosis process. Okay, I hope you understood all the introductory part of this process. Thank you.